Students, today the topic of our discussion is electron transport chain and oxidative phosphorylation, the ultimate steps for cellular respiration. Now, in our earlier classes, we have already learned that in glycolytic process that glucose is converted to pyruvate. One molecule of glucose is produced, two molecules of pyruvic acid. During this process, two ATP molecules were consumed in the first steps of glycolysis, and in the second stage, 4 ATP molecules along with 2 NADH molecules were produced. So, when we did the net energy balance, we have seen that net gain of 8 molecules of ATPs were there. In case of TCA cycle, we have also learned that how pyruvate is converted to acetyl-CoA, and when acetyl-CoA is formed, one molecule of carbon dioxide and one NADH is produced, and acetyl-CoA when enters to this mitochondria of the cell, we have seen that it is the 8 steps process, and during this completion, after the completion of this step, we have seen that 3 molecules of NADH, 1 molecule of FADH2 and 1 molecule of GTP is produced from 1 molecule of pyruvate. That means, when 2 molecules of pyruvates are being used in TCA cycle, we can get 6 molecules of NADH and 2 molecules of FADH2 and 2 molecules of GTP or ATP, and total gain of 24 ATP molecules were there. We have also seen that in this process some hydride ions are being formed, and when this cycle is further continued and it is further processed, it enters to the electron transport chain, and today we will be discussing on this electron transport chain. Now, if we see the total cell, suppose this is the intact cell, glycolysis is taking place in this cytoplasmic fluid of the cell. As soon as pyruvate is produced, it enters to the mitochondria. This is the mitochondria of the cell. Acetyl-CoA is produced and enters to this mitochondria. Mitochondria has got its 2 layers membrane, one is the outer layer, another is the inner layer. Now, outer layer is the protecting layer and inner layer is just folded inside the matrix, and it forms the cristae in the mitochondria. And when we see the structure of mitochondria, we can find that it has got the outer membrane, the inner membrane, and mitochondrial ribosome and matrices are there inside, and this infolded, that inner membrane is infolded inside and form the cristae-like structure. Now, if we see the outer membrane of this mitochondria, then we can find that the outer membrane is permeable to small molecules and ions because of the presence of the protein which is called the porine proteins. So, porine proteins are present which helps the transportation of some ions and small molecules through this outer membrane. Porins are transmembrane proteins that forms the channel in the outer membrane in the outer membrane of the mitochondria. The inner mitochondrial membrane is highly impermeable and requires specific transporter. That means, this inner membrane is highly intact and it is impermeable to any such ions or any soluble or insoluble molecules. Transporters are needed for transportation from inside to outside. The transporters specific for pyruvate, fatty acid, amino acid, ATP, ADP, phosphate and protons are found in the inner membrane of this mitochondria. And when we see the inside, this inside is the matrix. The mitochondrial matrix contains enzymes, that is pyruvate dehydrogenase, TCA cycle enzymes, beta oxidation and amino acid oxidations. And mitochondrial matrix is enclosed by two membranes. So, here the inner membrane and the outer membrane. So, this is all about this mitochondria, and why I am talking about this mitochondria so much, because this electron transport chain and the oxidative phosphorylation, today the topic of our discussion, what we will be discussing takes place in this mitochondria, and that is the reason why I am talking about the structure of mitochondria. Now, coming to this electron transport chain. Now, the NADH and FADH2 formed during glycolysis, beta oxidation and TCA cycle give up their electrons to reduce molecular oxygen to water. That means, this NADH and FADH2, we have already seen that NADH is formed in glycolysis, NADH is also formed in TCA cycle, FADH2 is also formed, and whatever these high energy molecules, they give up their electron to reduce this oxygen molecules and it converts to water molecule. Electron transfer occurs through a series of protein electron carriers, the final acceptor being oxygen. That means, this is the oxygen which is the final electron acceptor. The pathway is called electron transport chain. Electron transport chain takes place in the inner mitochondrial membrane. So, here in this portion, the electron transport chain is taking place, where all the electron carriers are present. The function of electron transport chain is to facilitate the controlled release of free energy that was stored in reduced cofactors during catabolism. Now, here if we see the electron carriers which further help to complete this electron transport chain, mainly composed of 4 different complexes: complex 1, complex 2, complex 3 and complex 4. This complex 1 is NADH dehydrogenase, complex 2 is succinate dehydrogenase, complex 3 is cytochrome BC1 complex and complex 4 is cytochrome C oxidase. Now, let us come one by one to this each complexes which help in electron transport chain. Now, at a glance, if we see that how this electron transport chain is, see here these different complexes which are there. So, see this is the matrix side of this mitochondria and this is the intermembrane space, that means the space between the space between the inner and the outer membrane. So, this is the intermembrane space of this mitochondria. So, if we are taking the view like this is the matrix side and this is the intermembrane space of mitochondria followed by the outer membrane outside, and if we see this cross sectional view, then we can find that the arrangement of complex 1, complex 3 and complex 4 are there, whereas complex 2 is present in the inner mitochondrial membrane, as I have already discussed that the succinate dehydrogenase, the enzyme which is mainly responsible for transferring the electron from complex FADH2 to the electron transport chain, that is the ubiquinone, is present in the inner mitochondrial membrane. That means we can say that complex 1, complex 3 and complex 4 are in one side and complex 2 is present in the inner mitochondrial membrane. Now, here followed by, and when it is getting completed complex 4, you see this oxygen molecule is getting this electron and it converts this oxygen to water molecule, and then this ATP synthesis is taking place, and that means when this electron transport chain is getting completed, then ATP synthesis is coupled up. That means this oxidation and phosphorylation is simultaneously taking place within the cell, and thus it is called oxidative phosphorylation process. With the completion of oxidative phosphorylation, we are getting the net production of ATP in the cell. Now, coming to this complex 1. Now, when we are talking about this complex 1, this complex 1 is NADH coenzyme Q oxidoreductase. Now, when we are talking about this complex 1, we can find that this NADH dehydrogenase is playing a significant role as far as this complex 1 is concerned. Now, what it does? This NADH dehydrogenase oxidizes this NADH. At the same time, it transfers this electron to ubiquinone, the ultimate electron acceptor, and at the same time it pumps 1 proton per 1 electron. So, this is the function of NADH dehydrogenase. Now, if we see the structure of this particular enzyme, we can find that this is the enzyme which has got two sides. One is the hydrophobic horizontal arm, which is buried inside the membrane, and another vertical arm contains the peripheral membrane proteins of the complex, which is projected towards the matrix. So, this is the structure. It is the L-shaped enzyme protein which helps in the catalytic activities. Now, here is the binding site. When NADH is coming and it is donating its electron to this particular enzyme, it immediately takes up and it starts its function. How it functions? There are different helping hands, that is the coenzymes which are present inside this particular complex 1, and these coenzymes are iron sulfur non-heme protein, FMN and coenzyme Q. This iron sulfur non-heme protein is one of the cofactor, that is otherwise known as FES complex. flavomononucleotide is another cofactor, and coenzyme Q is the another cofactor. These three cofactors are helping to transfer the electron from NADH to the ultimate site, that is coenzyme Q, that is ubiquinol. Now, here if we see this transmembrane space and this matrix, see NADH is donating its electron and it is getting transferred. At the same time, we can find that some protons are getting pumped from inside matrix to the outside of this intermembrane space. Now, see here NADH is giving this electron to flavomononucleotide and it is further passing to this FeS complex, that is iron sulfur protein. Ultimately, it transfers this electron to ubiquinol and it forms ubiquinol QH2. When this process of transfer of electron is going on, at the same time these protons are getting pumped out from inner matrix to the intermembrane space. Now, see here this NADH ubiquinol oxidoreductase, that is NADH dehydrogenase, transfer this electron from NADH to Q, that is ubiquinol. NADH transfer 2 electrons as per hydride ion to flavomononucleotide. Electrons one at a time are passed through the complex 1 to ubiquinol via FMN and iron sulfur protein, and in this way this function of complex 1 is getting completed. Now, this electron has come up to this ubiquinol and ubiquinol is converted to ubiquinol, that Q becomes QH2. Now, coming to this complex 2, as I have told you, the location wise that complex 2 is in the inner mitochondrial membrane, as this succinate dehydrogenase, this enzyme which is helping to transfer this electron in complex 2 is present in the inner mitochondrial membrane. It accepts the electron from the succinate and catalyzes the reduction of Q, that ubiquinol, to QH2. FAD of complex 2 is reduced in a 2 electron transfer of a hydride ion from succinate. Complex 2 does not contribute to proton gradient, but supplies electron from succinate. That means, here in case of complex 1, if you remember, here this proton gradient is from this pumping of this proton from inside matrix to this outside, this intermembrane space is taking place. But here in this case, only the transfer of electrons are taking place, and here also the ultimate acceptor of electron is ubiquinol and it is converted to ubiquinol. So, QH2 is the ultimate, this ubiquinol is the ultimate acceptor of electron. Now, in this way this FADH2 and NADH is donating the electron to the electron transport chain. It is further carried over to complex 3. In complex 3, it is the ubiquinol cytochrome C oxidoreductase enzyme which is playing a significant role, and it transfers this ubiquinol to cytochrome C, and here when it transfers this electron, it goes via the different enzyme cofactors like cytochrome B, ferrous iron protein, cytochrome C1 and ultimately it reaches to cytochrome C. Now, the transfer of electron to cytochrome C from ubiquinol is taking place. Oxidation of 1QH2 ubiquinol is accompanied by the translocation of 4 hydrogen ions across the inner mitochondrial membrane. 2 hydrogen ions are from the matrix, 2 from ubiquinol, resulting in a net transfer of 4 hydrogen ions from matrix side to intermembrane space. Now, when it comes up to the cytochrome C, this cytochrome C is accepting this electron, then it catalyzes a 4 electron reduction of molecular oxygen. Now, this sources of electron is cytochrome C. This cytochrome C is further donating this electron via this copper complex, and then it comes to cytochrome A3, and ultimately it reduces this oxygen to water molecule. During this process, these hydride ions are also getting pumped out from the matrix side to the inner mitochondrial matrix. So, translocation of hydrogen ions into the intermembrane space is taking place. Iron atoms, that is hems of cytochrome A, and copper atoms are both reduced and oxidized as electron flows, and transfer of 2 hydrogen ions from the matrix side to the intermembrane space is also taking place simultaneously. Now, if we see the flow of electron during this electron transport chain, we can find that we can sum up what we have learnt till now, that NADH is donating the electron to complex 1. From complex 1, ultimately it is coming, the acceptor of electron is the ubiquinol. Ubiquinol is transferring this electron to this complex 3. Here FADH2 is also transferring the electron to ubiquinol, and ubiquinol is once again transferring to complex 3. From complex 3 it comes to cytochrome C, cytochrome C to the complex 4, and complex 4 here this oxygen molecule is getting reduced and water is produced. And here during this process, you see in complex 1 these protons are being pumped out. In complex 3 also these protons are getting pumped out, and complex 4 the protons are getting pumped out. We do not see any proton pumping in case of complex 2. Now, if we sum up this, the particular studies what we have learned, that then I have already told you that 1 molecule of NADH is equivalent to 3 molecules of ATP. Now, how it can be proved? So, see here this NADH. NADH is donating its electron, and the possible sites of energy conservation in respiratory chain is this: ATP and PI is getting converted to ATP and water molecule. So, this is the first molecule of ATP which is being generated during this electron transport. Now, when this chain is continuing, FADH2 is from the complex 2, we have seen that they are also contributing this electron, they are also donating this electron, and ultimate electron acceptor is the ubiquinol, and this Q is accepting this electron. Now, you see here it is coming from the inner mitochondrial membrane. Now, when it enters to this particular electron transport chain, by this time this NADH, which donating its electron, already produced one molecule of ATP. Now, this chain is continuing, and when it is going through the complex B and C, cytochrome B and cytochrome C, the second molecule of ATP is getting generated, and when it is coming to A and this molecular oxygen, another molecule of ATP is being synthesized. So, we can tell that one molecule of NADH is producing 3 molecules of ATP, and one molecule of FADH2 is producing 2 molecules of ATP. So, this is the production pattern of ATP from one molecule of NADH and FADH2. Now, if we couple up, this is as far as electron transport chain is concerned. Now, when we are coupling up this electron transport chain and oxidative phosphorylation, then we can find that energy is released when electrons are transported from higher energy, that is NADH or FADH2, to lower energy oxygen. The energy is used to phosphorylate ADP. Coupling of ATP synthesis to NADH or FADH2 oxidation, at the same time phosphorylation is also taking place in the same place, and this process is called as oxidative phosphorylation process. Oxidative phosphorylation is responsible for 90% of the total synthesis of ATP in the cell. If we see the energy, the proton pumping during this electron transport chain, then we can find that there is the chemiosmotic theory which is there, which is established by the scientist Mitchell, and it is one of the theory which explain the mechanism of oxidative phosphorylation. When electrons are transported along with the component of electron transport chain, the accompanying protons are released. The part of the free energy harvested during the electron transport chain is used to pump the proton out of this mitochondrial matrix. The resulting uneven distribution of the proton generates the pH gradient and a charge gradient across the inner mitochondrial membrane. Now, here what is happening? When this process, electron transport chain process is going on, if this is the structure of mitochondria, protons are being pumped out from matrix to the intermembrane space, and here what is happening? The concentration of this H plus ion is more in this particular place than what is there inside. So, in this way one gradient is formed. That gradient is either electrochemical gradient or the pH gradient, which is formed inside the matrix and the intermembrane space. The electrochemical potential energy generated by this gradient is called proton motive force. The return of protons to this mitochondrial matrix is coupled to the ATP synthesis. Now, you see, now this is the electron transport chain. Say protons are getting pumped out from inside matrix to the outside. This is complex 1, complex 2, complex 3. Electrons are being transferred and oxygen is reduced to water molecule. Now, these protons are being pumped out of this inner mitochondrial membrane to the intermembrane space. Now, when it is further coupled to the ATP synthesis, the enzyme ATP synthase is playing a significant role. ATP synthase has got two moieties, one is the F0, another is the F1. And when this ATP synthesis is taking place inside this matrix, then protons are being pumped in the matrix and ADP and PI is phosphorylated and it forms this ATP molecule. And this way, this concentration difference, that when we are considering this electrochemical gradient, the concentration difference is because of the more proton on the outside, and this factor is the concentration difference is this more protons on the outside by a factor of 10. The charge differences are because of these protons are positively charged, which are more on the outer side than this inner mitochondrial matrix. So, there is more protons on the outside surface, that means more positively charged on the outside and negatively charged the inside. Now, if we come to this ATP synthase and if we learn little more about this particular enzyme, we will find that this enzyme is a very interesting enzyme. You know, ATP synthase, it is the proton dependent ATP synthase and it uses the proton gradient to synthesize ATP. It is the multi-subunit transmembrane protein and molecular mass of this particular enzyme is around 450 kilodalton. As I have already told you that it has got two moiety subunits, one is the F0, another is the F1. F0 is the water insoluble transmembrane protein and it has got 8 different subunits. F1 is the water soluble peripheral membrane protein and it has got 5 different subunits, among which beta subunit is actually participating, it is catalyzing a significant role as far as ATP synthesis is concerned. Now, this beta subunit is also very interesting. Now, here if we see this beta subunit, that is in the F1 portion, F0 and F1, it is the F1 portion of this particular ATP synthase. Now, if we see this beta subunit, we can find that it has got L form, O form and T form. 3 catalytic sites are there. What are the function of these 3 catalytic sites? The L form is the substrate binding and catalytic substrate binding site, but catalytically it is inactive. O form is the open form and it has a very low affinity for the substrate. That means it does not have any affinity for substrate binding. And T form is the tightly binding and ATP with ATP, that means it has got a very strong affinity for ATP and it is also active. Now, this if we are symbolizing this L form, T form and O form, then we can find that this L form is having, this is the substrate binding affinity, but catalytically it is inactive. That means it can only bind the substrate, but it does not have any catalytic activity. This T form, it has got very strong affinity for its substrate binding and also it is active in nature. So, it can bind this ATP molecule to this, and O form is does not have any substrate binding affinity, and it is the open form. So, it is present in this. Now, when pumping of proton starts, the beta subunit gradually changes its conformation, and this is the beauty of this mechanism of this particular beta subunit of ATP synthase. Now, here when this ADP and PI is getting binded with the L side, and when electron flow starts, then it starts changing its orientation. This conformational changes takes place. So, L state, it binds the substrate ADP and PI. So, energy dependent conformational changes when takes place, this L side become O side become T side, and T side becomes L side, and this way it starts changing its conformation, and in the T state formation of phosphoanhydride bonds are taking place, resulting in the formation of ATP molecule, and as soon as the O state is coming, and this O state is releasing this ATP molecule, and in this way ATP synthesis is taking place in this particular with this help of this particular enzyme ATP synthase. Now, if we see, this is the ATP synthase, this is the F0 form, this is the F1, and when we see this proton concentration, the gradient outside of this of the intermembrane is more than the inner matrix membrane, then protons are being pumped in the inside in the matrix zone of this particular mitochondria, and during that time ATP synthesis is going on. That means, when these protons are getting pumped in and concentration of protons inside is less, outside is more, ATP synthesis is taking place. Whereas, when protons are getting pumped out during this electron transport chain, the ATP hydrolysis is taking place, and when ATP is giving rise to ADP and PI, and system is getting energy, and protons are being pumped out from inner mitochondrial membrane to the intermembrane space, and in this way the ATP synthesis and ATP hydrolysis are taking place simultaneously along with the electron transport chain. And now, if we see sum up this electron transport chain and oxidative phosphorylation, then we can couple up both the things, that when ATP is converted to ADP, protons are being pumped out from inner mitochondria matrix to the intermembrane space. When electrons are getting flowed, the protons are being pumped out from inner mitochondrial membrane to outer mitochondrial membrane, and in stage 3 this ADP and PI is once again synthesizing ATP, electrons are flowing and protons are being pumped in the matrix, and ATP synthesis is taking place, what I have already mentioned in electron transport chain coupling with oxidative phosphorylation process. Now, what are the minimum requirement for carrying out this oxidative phosphorylation in the mitochondria? Here the supply of electron through the electron transport chain is essential, and electrochemical gradient, that is the potential difference between the matrix and intermembrane space, is essential. ADP, PI and O2 should be there, and active ATP synthase system in the inner membrane of mitochondria is very essential, and most important part is an intact inner mitochondrial membrane, and this is playing a significant role. If there is any breakage or anything, there will not be any gradient formation resulting in the discontinuation of the oxidative phosphorylation process. If we see different inhibitors of electron transport chain and oxidative phosphorylation, we can find that in electron transport chain that rotenone, carboxyl, antimycin A, cyanide, azide, carbon monoxide, etc. are the inhibitor of this electron transport chain. Here in the plain straight electron transport chain, we are considering complex 1, complex 3 and complex 4. We can see that complex 1 is getting inhibited with rotenone. Antimycin A is inhibiting this portion, and further continuation of this chain length is getting stopped, and in the aid cyanide or azide, they stopped in this cytochrome AA3, and here it is getting stopped. And similarly, ATP synthase is also getting stopped by a series of these inhibitors which inhibits the enzyme. The role of uncoupler to inhibit oxidative phosphorylation is already there, and they uncouple, mainly they uncouple the electron transport chain from oxidative phosphorylation. The electron transport chain remain intact and electrons are transferred to oxygen generating water. However, uncoupler carry the proton across the mitochondrial membrane, making it leaky for hydride ion. The pH of the electrical gradient is not generated and ATP is not getting synthesized. In the presence of an uncoupler agent, energy released via electron transport chain is converted into heat. The mechanism is used by hibernating animals to stay warm in the winter, since they do not need ATP for anabolic process while they are resting. And there are different uncouplers, and most important natural uncoupler is the thermogenins or some uncoupling proteins, and for synthetic uncoupling reagent, that is 2,4-dinitrophenol is one of the potent inhibitor uncoupling agent. Now, as I have told you that inner mitochondrial membrane is not permitting anything for going out of this system. Now, when we are talking about this electron transport, this electron shuttle system, it helps in the transportation of NADH to the mitochondria. And when we are talking about this mitochondrial membrane, it is impermeable to NADH, and thus transportation, because flow of electron is essential. So, whatever this NADH or FADH2 is produced in the cytosol of the cell is being transported inside this mitochondria, and for this particular activity, the two shuttle systems are existing. One is the glycerol 3-phosphate shuttle, another is the malate aspartate shuttle. In case of glycerol 3-phosphate shuttle, it functions mainly the skeletal muscle and brain. As we have already learnt that NADH on the cytoplasmic side is oxidized to NAD with coupled reduction of dihydroacetone phosphate and glycerol 3-phosphate. The oxidation of glycerol 3-phosphate back to this dihydroacetone phosphate is catalyzed by a mitochondrial membrane bound isozyme, that is the glycerol 3-phosphate dehydrogenase. This enzyme is there, and this oxidation is coupled to a reduction of FAD, the prosthetic group of the mitochondrial enzyme, to FADH2, and this FADH2 which is now produced is transferring this electron to the ubiquinone, and it is converting this ubiquinone to ubiquinol, and this way it is helping the electron, the supply of electron to this electron transport chain. And thus in muscle and brain, even though 2 NADH are produced by glycolysis, actually 2 FADH2 are available for the entry into the electron transport chain. Another very interesting shuttle which is taking place in the electron transport chain is the malate aspartate shuttle. In the cytosol, electrons are transferred from NADH to oxaloacetate, forming NADH and malate. The enzyme is malate dehydrogenase, which is one of the very important enzyme, as I have already discussed in TCA cycle. Malate can easily enter mitochondria. In the mitochondria matrix, the malate is oxidized to oxaloacetate by the enzyme of this TCA cycle, that is the malate dehydrogenase. This is coupled to the reduction of NAD to NADH. Oxaloacetate is converted to aspartate by accepting the amino group from glutamate in the reaction catalysis by this amino transferase. So, this aspartate is converted to oxaloacetate is converted to aspartate. So, this is the oxaloacetate which is getting converted to aspartate. Oxaloacetate is converted to aspartate, and aspartate as soon as it is produced, so it is coming to the cytosol, and here in presence of this aminotransferase, once again oxaloacetate is being formed and it is converted to malate, and malate is going inside this mitochondria, and in this way TCA cycle is getting continued, and the glutamate which is there, this glutamate is donating its amino group and alpha ketoglutarate is formed, and this alpha ketoglutarate is once again coming to the cytosol of this particular cell, and it is converted to glutamate, and this way this shuttle is going on. The aspartate readily crosses the mitochondrial inner membrane into and enter the cytosol. In cytosol, aspartate donate its amino group to form oxaloacetate. Thus, in heart and liver, electron transfer from cytosol to mitochondria, and it does not involve any expense of energy, and this is the shuttle system in case of this electron transport chain and oxidative phosphorylation. And here if we see the total energetics, how many ATP molecules are being produced from glycolytic process, because this is the ultimate steps of respiration. It starts from glucose to pyruvate, and it enters to pyruvate is converted to acetyl CoA, enters to TCA cycle, and then it flows to electron transport chain followed by oxidative phosphorylation, and here the cellular respiration is getting completed. So, if we see the total this process, we can find that in case of glycolysis, the first step 2 molecules of ATP is being consumed, so minus 2 molecules of NADH is produced, 4 molecules of ATP is produced. In case of pyruvate dehydrogenase, 2 molecules of NADH is being produced. In TCA cycle, 2 ATP, 6 NADH and 2 FAD molecules are produced. In glycerol 3-phosphate shuttle, 2 molecules of NADH is being consumed and 2 molecules of FADH is produced, FADH2 is produced. So, total gain, if we say, we can get that 4 molecules of ATP, 8 molecules of NADH and 4 molecules of FAD is produced, which is giving this 4 molecules of ATP, 24 molecules of NADH, because one NADH is equivalent to 3 molecules of ATP, we have already seen. So, 8 molecules of NADH means 24 molecules of ATPs, and 4 molecules of FAD means 8 molecules of ATP. So, altogether we can see that there is a net gain of 36 ATP molecules when the intercatabolic process, the cellular respiration process is getting completed. Now, in conclusion, what we can tell, that virtually all energy released from the oxidation of macromolecule is made available in mitochondria as reducing equivalent, that means the hydride ions and the electrons. These are funneled into the respiratory chain, where they are passed down a redox gradient of carriers to their final reaction with oxygen to form water. The redox carriers are grouped into respiratory chain complexes in the inner mitochondrial membrane. These use the energy release in the redox gradient to pump protons to the outside of the membrane, creating an electrochemical potential across the membrane. Spanning the membrane are ATP synthase complexes that use the potential energy of the proton gradient to synthesize ATP from ADP and inorganic phosphate. In this way, oxidation is closely coupled to the phosphorylation to meet the energy need of the cell. So, with this, I think we have already learned the cellular respiration of any living cell. So, how this catabolic mechanism is going on inside the cell, and how this glucose to oxidative phosphorylation is continuously carrying out in any aerobic system. With this, I think we have already learned. Now, with this, I think this respiration in case of any aerobes are already discussed in details. In my next class, we will discuss some other metabolic activities which are going on in any living system. Thank you very much.